Okay, today's lecture, if you guys do not have any questions, then today's lecture is chapter 6. It is matters of sex. So far, we've been talking about traits which are located on our autosomes. That means chromosome pairs 1 through 22. So when it's an autosomal trait, irrespective of whether it's recessive or dominant, we know that it does not differentiate between whether the trait is being inherited by a daughter or a son. So the gender or the sex of the offspring does not matter in case of autosomal inheritance. But when a trait is located on our sex chromosomes, that means chromosome pair 23, then whether the trait is going to a daughter or a son, it becomes different because the sex chromosomes are different in case of a male and a female. So today's lecture is going to be on traits that are somehow directly or indirectly linked to our sex chromosomes or the sex hormones, testosterone and estrogen. So we know that in human beings, maleness or femaleness, gender or sex is determined genetically at the time of conception. As soon as a zygote, an egg gets fertilized, precisely at that moment, the gender genetically is determined. So if an X chromosome, if the 23 chromosomes from the dad contained an X chromosome, then when it fuses with the 23 chromosomes from the mom, the resulting baby is going to be a female because the X came from dad. But if the sex chromosome from the dad, instead of an X, contained a Y, not X. So as soon as it fused with the female ovum, it would be a male baby genetically, okay? So the maleness or the femaleness is determined by the sex chromosome that is being carried by the spermatozoa. Now this is genetic sex, genetic gender. We know that another level of sexual identity, because sexual identity is a very complicated process. It is not only based on what your chromosomes say, but also the influence exerted by sex hormones. The group of male sex hormones are known as androgens, the primary being testosterone, and in females it is estrogen. And then on top of it, the third layer, there is the effect of the society around all of us, and there are psychological components also. So all of these, this is a multi-layered process, the sexual identity or the gender identity. So we'll start, and as far as this lecture is concerned, we will be talking more about the genetic sexual identity. So if it's an XX, we will consider it as a female, and if it's an XY, then we're going to consider it as a male, irrespective of what the individual's external gender identity, whatever the individual identifies himself or herself as. So when a zygote is formed, when the egg gets fertilized, by the 5th week of embryonic development, remember up to 7th or 8th week, it's still an embryo. After that, it becomes a fetus. So by the 5th week, actually the developing embryo, it develops two kinds of gonads. Gonads means testes and ovaries. So the embryo, which is only 5 weeks old, has both ovaries and testes, precursors of ovaries and testes, and there are two sets of ducts. So the embryo can go either way. The embryo can either become a male or a female, depending on if the embryo contains a Y chromosome or not. So the presence or absence of Y chromosome will now influence the deterioration or the atrophy of one set of sexual organs. That means the individual has both, the embryo has both male and female reproductive parts, and then depending on whether this male gene, SRY gene, SRY gene, which is located on Y chromosome, if the SRY gene is present in the embryo, then those precursor parts that were supposed to become the ovaries, those will deteriorate and the embryo will go on to develop as a male. While if the SRY gene is absent in the embryo, that means the Y chromosome is not there, then those reproductive parts which were supposed to be testes, they will atrophy, they will deteriorate. So you see here, this is the, in this figure, this is the embryo with both sets of gonads and both sets of tubes. Those precursor tubes are known as Mullerian and Wolffian. Wolffian is for the male part, Mullerian is for the female part. Now, depending on whether the SRY gene is present or the SRY gene is absent and the SRY gene is on Y chromosome, it will determine which of these two reproductive organ sets will atrophy, will deteriorate, and which one will persist. So if the SRY gene is present, then it will develop into testes and all the male reproductive parts, while if the SRY gene is absent, the embryo is going to develop all the female reproductive organs. So sex chromosomes genetically determine the gender. In case of human beings, females or women are the homogametic sex. Homo means the same, gametic means producing gametes. So in case of human beings, females are XX. That means the two same type of sex chromosomes. While in case of men or males, they are heterogametic. Hetero means different. So that's why they have X and Y, and that's why they produce two different types of gametes or two different types of sperms. Sperms can either carry an X chromosome or carry a Y chromosome. An ovum will always carry an X chromosome, so homogametic. But this is not true for all the different animal species. We have seen that in birds and in snakes, okay, in lizards, snakes, males are actually homogametic and they are, it's denoted by ZZ, while females are heterogametic, ZW. So not all the, animals they show the same pattern. Now let's look at X and Y chromosomes, because these are the two chromosomes which determine maleness and femaleness. So if you see, this figure shows you an X versus the Y chromosome. You see how small the Y chromosome is. So a female will have two of these big X chromosomes, while a man would have an X and a Y. So going back, the X chromosome is much bigger, and because it's bigger, it has a lot more genes. There is more space for genes. It has more than 1500 genes, while Y chromosome contains roughly 231 genes. And the Y chromosome is sometimes a big mystery, because a lot of the segments, the DNA segments, are palindromes. Palindrome means a word or a phrase that you can read the same way forward and backwards. For example, if you write madam, M-A-D-A-M. So forward also it's madam, and then backward also it's madam. Or race car, forward and backward. You can also have a full sentence if you allow for some capital letters and punctuations and other things. If you just don't take them into consideration, then you can even have full sentences which are palindromic, like Abel was I saw Elba, so something like that. So those are sentences that are palindromic. And when we say palindromic DNA, means the DNA sequence could be, let's say it starts with an AA, then G, and then the ending also you could go AAG, you go backwards and it would be the same. And when DNA looks palindromic, then there is always a scope for error, because DNA has to duplicate, and whenever there are palindromic sequences, then the scope for error increases. So a lot of the DNA segment in Y chromosome is palindromic. Now let us try to understand Y chromosome. This is what the Y chromosome looks like. If you take the Y chromosome, only 5% of the chromosome contains pseudo autosomal regions, while 95% of this chromosome is mainly associated with male specific genes, or it's known as male specific region, MSY. So this 5%, which is pseudo autosomal, pseudo means fake, false. Autosomal is, autosomal. So this part of the Y chromosome is pretending to be like an autosome, but it's not. So that's why it's a pseudo autosomal region. And the genes that you see there are mainly PAR1 and PAR2, okay? While the 95% of the Y chromosome contains genes which are responsible for masculinity, which are responsible for maleness. So you will see the SRY gene there. You will see another gene called AZF there, okay? Azoospermia factor. So those are all the MSY gene. Now let's talk about the SRY gene. The SRI gene makes all genes, most of the genes make protein. So this gene makes a transcription factor. That is this gene's end product. Transcription factor means you're going to learn about transcription subsequently in this semester. Whenever genetic information passes from your DNA, from the DNA to the protein, DNA is the king, protein is the worker. And in between there is an RNA that is taking the information from the DNA to the protein in the cell. And the process by which the RNA is formed from DNA is known as transcription, and we need transcription factors. So the SRI gene helps in making those transcription factors, and it stimulates expression of other genes. So it is helping other genes that are responsible for male development. Then in case of males, these two hormones, which are very much required for development of secondary sexual characteristics, like testosterone and dihydrotestosterone, DHT, these hormones are also secreted because of the presence of this particular gene. So this gene is going to help in a pathway of excitation of other genes, and all these other genes are directly or indirectly related to development of maleness. So this figure explains it very clearly. You see, this is an ovum which contains an X chromosome, another ovum containing X chromosome. Ova will always have X chromosome. And here is a sperm which may contain Y, while there's another sperm containing X. So if this sperm fertilizes this ovum, then this is the fertilized egg, which would become a female, XX. While this sperm fertilizing this ovum would make it an XY zygote. And this XY zygote would have the SRI gene. This one would not have the SRI gene. So under the influence of SRI gene, the testes will develop. Because of the absence of SRI gene, ovaries will develop. So remember that ovaries develop not because of a specific gene. Ovaries develop because of the absence of SRI gene. And then when testes develop, then this leads to the development of other things inside the testes as well as secondary characteristics. Now, we're going to start with abnormalities or diseases or syndromes, genetic problems. What could be some abnormalities in sexual development? One of the abnormalities is pseudo-hermaphroditism. So let's try to break up this word. It's a really long word. Pseudo means fake. Hermaphrodite. Hermaphrodite means an organism that exhibits both male and female sexual structures. For example, earthworms. Earthworms are hermaphrodites. They are male on one end and female on the other end, so they can self-fertilize. So sometimes in a human being, there could be a false pretension of being a hermaphrodite. So this is presence of male and female structures, but this individual has the structures at different stages of life. And this could be due to genetic abnormalities. And some of the genetic abnormalities which could cause these are the androgen insensitivity syndrome, 5-alpha reductase deficiency, and congenital adrenal hyperplasia. Now, these are difficult things. In order for us to understand it easier, let's try to see this figure here. It starts with the SRI gene, okay? So during embryonic and fetal development, the SRI gene, let's say this is supposed to be a male embryo. So the SRI gene turns on and the testes have developed. So the female structures will degenerate. And now because the SRI gene is on and the testis has developed in this embryo, now what are the next steps for development of maleness? In the embryo, there is cholesterol. Remember that for all of our steroid hormones, testosterone, estrogen, cortisol, all of our steroid hormones are made from cholesterol. Cholesterol is the precursor molecule. No testosterone will be formed in our body. No estrogen will be formed if our body does not have cholesterol. So cholesterol, which was so far just basic cholesterol, as soon as testis has become functional, now this cholesterol is going to develop chemically. It is going to change into or form testosterone. And then this testosterone will also undergo some more chemical reactions and form another male hormone, dihydrotestosterone. So if an individual is healthy and normal, this is how it should go. But there could be problem at different steps. So the green arrows indicate where mutations can disrupt normal sexual development, and then the red, whatever is written in red, is the disease that is caused if this is what happens. So let's say a mutation occurs in the pathway here. The genes that were helping the cholesterol to be changed into testosterone, they get mutated. So now cholesterol is not changing. Testosterone is not getting formed. If testosterone is not getting formed, what is the disease now that is occurring because of this genetic disorder. It is called congenital adrenal hyperplasia. Okay, now imagine what will happen if testosterone is not getting formed. So what would have happened if testosterone had formed? If testosterone is formed, it passes through the plasma membrane of cells in the developing cells, and it causes the formation of internal male structures. So there are genes, and there is the influence of this hormone which helps in the development of the male reproductive system. So the epididymis is a comma-shaped structure where sperms mature. The ductus deferentia is the tube that is going to carry the sperm from the testes up to the urethra. The seminal vesicles are accessory organs. The ejaculatory duct is the path that carries the sperm out. So all of these structures will form only if testosterone is present in an optimal amount. So if the testosterone does not get formed because of this disease, none of these structures will develop. Okay, so and if there are receptors here which on cells that welcome the testosterone. What if there is a mutation that damages these receptors and the receptors are not formed? Then there's another disease. It's called androgen insensitivity syndrome. That means this individual is making testosterone, but the cells are not receiving the testosterone, and the end result, again, these structures will not get formed. Now let's say this part was still normal, and this part from testosterone to dihydrotestosterone is not getting done. Why? Because of a mutation here which stops this. This disease would be known as 5-alpha reductase deficiency. Now, what was the function of dihydrotestosterone? Again, it was going into the cells, and there were receptors. And when the dihydrotestosterone is working on the cells, it leads to the formation of the external male structures like the penis, like the scrotum, like prostate gland. So if dihydrotestosterone is not there, or if the receptors are not working, these structures are not going to be formed. So in a nutshell, what we can say is, if something goes wrong in this entire pathway, genetically, this embryo might be a male. That means if you look at the chromosomes, this embryo has an X and Y, but phenotypically or externally, this individual in the infant stage will grow up as a female. This individual will think that I am a female because of the absence of male reproductive structures. So that is, there are sexual abnormalities. Now let's look at homosexuality, which means preference for members of the same sex. Now, homosexuality has existed, for thousands of years in all the cultures, and not only in human beings, but in other animal species as well. And homosexuality, or even heterosexuality, any kind of sexuality, is not just genetic. As I said before, it's a complex input. It's an interaction between the genes and environment. Research is going on in this area. It is still controversial, but people have used identical and fraternal twins to study this. How much is the genetic contribution? How much is the environmental contribution? So as I had said in one of the previous slides, sexual identity is a multi-layered process. So you see here, what are the different timings for the different types of sexual identity? Your chromosomal or genetic identity occurs at the time of fertilization. As soon as fertilization has occurred, genetic identity has been sealed there. It's either an XX or an XY. Gonadal sex means the precursor sex start developing 6 weeks, between 6 to 8 weeks, there is the development of the external and internal reproductive structures. So this is gonadal or phenotypic sexual identity. Then comes the third or the fourth layer, gender identity, which can start from childhood or early adulthood, where there's a strong feeling of I'm a male or I'm a female. So that is your gender identity. And finally, sexual orientation could be whether this person is being attracted to a person of the opposite sex or the same sex. And this could again also start from childhood. So this is a multi-layered process, and there are different layers which would help the person get his or her sexual identity. Sex ratios. Sex ratios means in a population, the proportion of males to females. So we count the total number of males, and then we count the total number of females. We divide these two numbers and then multiply it with 1000. And when we talk about sex ratio, we have to be very specific if we're talking about primary sex ratio, secondary sex ratio, or tertiary sex ratio. Primary means at conception, and at conception probability says 50%, because it's like tossing a coin. 50% of the times you'll get a male, 50% of the times you'll get a female. Now secondary sex ratio at birth ideally should again be 50%, but unfortunately in some countries, in certain countries, in certain cultures, because of a preference for a male child or a female child, people illegally or legally, whatever, they get rid of, they abort the female fetus, or vice versa. Okay, it could be other one also. So the secondary sex ratio differs, or sometimes it could be the natural variation as well. While tertiary sex ratio is at maturity, how many survived? Okay, how many of the males or the females died? And so what is the sex ratio at adulthood? And again, ideally this should be 50% males, 50% females, but this does not happen. All over the world, we see that the number of women in a lot of countries is going down. Okay, it's a very scary situation. So Y-linked traits, that means traits or characters that are located on Y chromosome. These are transmitted from male to male because it is Y chromosome. Y chromosome is never going to a female. So it is from grandfather to dad, from dad to son, and these are some genes on the Y chromosome. The SRY gene, the testis determining factor TDF, ZFY, or the other genes. Okay, so we go to X-linked traits now. This is more complicated than Y. Y chromosome is small, not much, and the inheritance is easy for Y chromosome. X-linked traits can go to son, can go to daughter, because remember, men also contain one X chromosome. So what are the possible genotypes with the X-linked traits? So let us first consider in case of women, okay? So women will always have two X chromosomes. We write it as capital X, capital X. Now let's say this is a wild type, okay, or this is a normal. So we write this as homozygous wild type female. Now, one of the X chromosome carries the mutant allele. So then this becomes a heterozygous female carrier, okay? And if both the X chromosomes carry the mutant allele, then this is a homozygous mutant female. So these are the three types. What about in men? When it's XY, well, in case of men, it's always XY, and this X chromosome could either carry the wild type version, or the mutant version. So when it is the wild type version, instead of calling it homozygous or heterozygous, because it is neither, we call it hemizygous. So this is a hemizygous wild type male and a hemizygous mutant male, if this is the mutant version. You see here, this is the female, this is the male, these are the two X chromosomes that she's carrying. So when you do the segregation of gametes, Mendel's first law, two X chromosomes get separated. In case of men, the X and the Y get separated, and this is all the possible, when you draw a Punnett square, all the possible combinations of gametes. So traits that are present on X chromosomes, these could also be recessive, or it could be dominant. So some of the X-linked recessive traits are color blindness, the inability to distinguish between red and green. So these people, they have difficulty getting driving license because they won't be able to see the difference between a red light and a green light. Hemophilia, which is the royal disorder of blood clotting, and these are both X-linked recessive trait. So recessive means, remember, for a recessive trait to be expressed, it needs double dose, where if there is any wild type dominant version along with it, this particular trait will not be expressed. So if you see, let me go back to the previous slide here. Think of this M as a recessive trait. If you see this slide, now this female, she does not have the mutant version. So she is perfectly healthy, homozygous wild type. Now this female has this one mutant version, but this is recessive. So will she show color blindness or hemophilia? She wouldn't. because this X is normal. This is carrying a dominant, healthy version of the allele. So this would be a female carrier. She might carry the mutant gene. She will pass it on to her offsprings, but she herself is not affected because she is simply female carrier. So for a female to be expressing this disorder, for a female to be colorblind, for a female to have hemophilia, what kind of genotype is required? This kind, where both X chromosomes need to have these mutant alleles, okay? So this particular female will show color blindness. But if you think of the males, this male has the healthy X gene, okay, the wild type. But this male has the color blindness, and there is no other X chromosome that is dominating or that has the wild type version. So for a male, there is only one X chromosome, and that X chromosome has the defective allele. So this male will actually express color blindness. This male will express hemophilia. So you see, when it's an X-linked disorder, if you compare the inheritance in a daughter versus a son, it's markedly different, because the daughter needs to have the defective allele in both X chromosomes, while for the son, a single X chromosome with this particular abnormal or defective allele would cause the son to be colorblind or to have hemophilia. This is a pedigree of the royal family of Europe showing hemophilia. You see here, these were, Queen Victoria I and her husband, and they were normal. It is at this stage where Queen Victoria of England, she got, she is the first ones. Suppose it is thought that a mutation occurred here, and then she passed it on to her offspring. Some of them were affected, some of them were carriers. So this son, who's Duke of Albany, he was affected. He had hemophilia. This is a daughter who was heterozygous, so she was carrying it. Another daughter was heterozygous, and this is how the disease has passed on to the Russian royal family, the German royal family, Spanish royal family, and this is the modern English royal family. This is hemophilia. Now, X-linked dominant inheritance. This could be a problem, because when it's dominant, then only in one copy it is sufficient to express the disease. So even in females, if only one X chromosome gets that, then that female will show the disease. And the effects would be, of course, much more severe in males. Now, whenever it's a dominant, there would always be a possibility that in the embryonic stage, it could be lethal. And so that those embryos would get spontaneously aborted. They won't even survive. An example of X-linked dominant inheritance is a patchy pigmentation disease. So whenever you have to solve genetic problems using X-linked inheritance, you need to follow these steps. They're very similar to how you would do any autosomal disorder, but keep in mind, if it's an X-linked trait, if it's a sex chromosomal trait, then it will be very important, because males are hemizygous, okay? Even one X chromosome would lead to expression of that trait. Now, all disorders may not be directly located on the X chromosome, and that is how they are affected. There are also other types of traits which are influenced by the gender of the person. One of the type of trait is known as a sex-limited trait. A sex-limited trait means those traits that affect a structure or a function of the body that is present only in males or in females. So the gene may be either present on the X chromosome or present on the autosomes. The gene may be present in both males and females, but in one of the gender, let's say in females, it is not expressed, okay, and it doesn't work. But in other gender, as soon as it is present in males, then it expresses itself. For example, the growth of beard or milk production in Maj cows. So these are traits, the genes for these traits are present both in male and female versions, but this gets expressed only in one gender. Or cryptorchidism, which is undescended testicles. So the genes are present in both, but since women don't have testicles, it's present only in men. So that is where the gene gets expressed. Now, the next category of trait is known as a sex-influenced trait. Sex-influenced trait means that trait where the phenotype is very strongly influenced by sex hormones, okay? So that means the allele behaves like a dominant allele in one sex, but it behaves like a recessive allele in another sex. And a very classic example is pattern baldness. So the gene for pattern baldness, okay, this is an autosomal gene. This is not even present on the X chromosome. So both men and women have this gene, but you see pattern baldness is more common in men. You rarely see it in women. And the reason is the baldness allele behaves like a dominant allele in males. So even if it is present, in single copy, it would still express itself, but it behaves as a recessive allele in women. So for it to be expressed in women, there needs to be 2 copies of those alleles. So that is why this is a sex influenced trait, and it is actually depends on the proportion of testosterone, the level of male hormones. Now, an interesting phenomenon occurs in women, which is known as X inactivation or equaling out sexes. If you look at any other autosome, there are pair of chromosomes, like chromosome pair 1, chromosome pair 2. So that means you have 2 alleles for each gene, right? So whatever allele is present on a certain region in one homologue of chromosome pair 1, the second homologue will also have the same allele. So if a gene is present on the X chromosome, if it's a woman, if it's a female, then the females would have 2 alleles for that particular gene on X chromosome. But what about men? Because men only have 1 X chromosome and the other chromosome is Y, and the Y is really small and it doesn't even have all the genes that X chromosome has. That means women end up getting double the amount. They have 2 alleles for each of those genes present on the X chromosome, but men don't have it. So that means the women would be like all super women and men don't have it. So nature balances this by causing inactivation of 1 X chromosome, which is known as X inactivation. Not only in human beings, in mammals, this inequality is balanced. How? 75% of genes on 1 X chromosome would get randomly inactivated in each cell. So as a zygote is formed, at the time of zygote formation, very quickly as the zygote is developing into 2 to 4, 4 to 6, like in 6 or 8 cell stage, one of the X chromosomes out of the 2, in each of those 8 cells would get inactivated. Which X chromosome? Would it be maternal X or paternal X? Is totally random, okay? And it's not, there's no guarantee that out of those 8 cells, in all of the 8 cells, the paternal X chromosome is only getting inactivated. No, okay? The 8 cells could have different X chromosomes getting inactivated. One cell has the maternal X being inactivated. The other cell has a paternal X being inactivated. But once the X chromosome has been inactivated, then each of those 8 cells, when they are dividing to form more cells, those cells will follow what the original cell, whichever chromosome was getting inactivated. And this inactive X chromosome is now known as a bar body. And you can see it under the microscope. It appears to be small and really darkly stained. So how does this affect X-linked genotypes, especially in case of women? So for homozygous X-linked genotypes, X inactivation has actually no effect, okay? Because both the X's had the same allele. So if one of them got inactivated, doesn't matter, the other X is going to show it. But for heterozygotes, okay, how does it affect the heterozygotes? Let's say the woman had 1 wild type allele on 1 X chromosome and 1 mutant allele on the other X chromosome, and then one of the chromosomes has got randomly inactivated. So for heterozygotes, it would cause expression of 1 allele, preferentially 1 allele over the other, because the other one that has become a bar body is not going to affect it. So how does it, in general, how does it affect the health of the women? Well, this doesn't affect the health if enough cells express the normal allele. However, if it has so happened that most of the cells where most of the X allele that got inactivated were the normal ones, and it is only the unhealthy, the disease carrying one that has remained active, that means the female will now show the disease because most cells contain that particular defective X allele. So this is X inactivation would, as I said, would occur very early when the embryo is still developing, 8 cell stage. And this inactivation happens not because of a change in the DNA sequence. Oh no, the DNA is not, altered in any way. These are known as epigenetic changes. Epigenetic changes means changes in DNA sequences without changes in DNA without changing anything in the sequence. So what happens usually in epigenetic changes are things get added to it from externally. So let's say a DNA sequence is there, and if you bring in a methyl group CH3, you add it to the DNA. Now the DNA looks different and it behaves differently. So this has been an epigenetic change, and epigenetic changes can totally change the gene product, the functional product of the DNA, and this is an epigenetic change. And the gene that causes this change is known as XIST, X-I-S-T. You see the full form here. This is the gene that encodes an RNA, and this would bind to and inactivate the X chromosome, right? So every cell in the body of female human beings, one X chromosome is inactivated. Now, what would happen at the time of meiosis inside the ovary when a new egg is being created? So does it stay like, okay, this chromosome is still inactive? Oh no, at the time of any germline synthesis, that means at the time of reproduction, whenever the stem cells in the ovaries are getting ready to make new oocytes, new ovum, this change gets reversed. It's an epigenetic change. It can be reversed. So it goes back to being reversed, and the ovum ends up having, again, it would have, each ovum would have a functional X chromosome, so that no ovum is there which has the par body. So that is how it works. So effect of X inactivation on phenotype. A female carrier who's having an X-linked recessive trait and it is expressing the phenotype would be known as a manifesting heterozygote. Why? Because phenotypically, or if you think of expression-wise, the female is behaving like a male because one of the X chromosome has become inactivated. So even if she was a carrier of a disease and enough cells in her body are expressing that abnormal allele on the X chromosome, then she is a heterozygote, but she manifests or expresses that disorder. And these are interesting studies. Studies have been carried out in women who were carriers of hemophilia or some other X-linked disorders. All of these are being done to study how X inactivation influences the development of sexual identity. Genomic imprinting is another epigenetic alteration. Remember, epigenetic means no change in DNA sequence. So here the phenotype of an individual would differ depending on if the gene is coming from the father or from the mother. And this genomic imprinting is usually done by DNA methylation. That means a methyl group comes and binds to the DNA, and this could either overexpress or suppress the expression of a particular gene. So you see here, this is the DNA sequence, the greenish, yellowish, all of these little things. You see these red things, these are methyl groups. So they will come and they will bind to DNA at specific spots, bind into the grooves, and they change the expression of the DNA. So let's see here, importance of genomic imprinting. I wanted to show you two examples right here. There's a particular mutation, okay? So that mutation, the deletion mutation, that means a part of a gene gets deleted in a certain chromosome, chromosome number 15. And an individual who inherits this abnormal chromosome will show some kind of genetic disorder. Now it is interesting that research has shown that individuals with chromosome 5 deletion can actually exhibit two different types of syndromes, two different types of disorders, depending on whether the deletion was inherited from the paternal side or from the maternal side. So the Prader-Willi syndrome, this is if the individual inherits the deletion from the father. And what are the symptoms? There's an eating obsession, metabolism becomes very slow. But if the individual inherited this chromosomal deletion from the maternal side, then this individual would not exhibit this syndrome, but would rather would have this syndrome, which is known as the Angelman syndrome. And this causes autism and intellectual disability. So it was the same chromosome 15 deletion, but depending on whether it was inherited from the dad or from the mom, the individual is showing two different diseases. Now there could be other causes of this disease as well, uniparental disomy, where parent gets two copies of the chromosome from the same parent. So you see here Prader-Willi and Angelman syndrome, same chromosome 15 deletion, paternal and maternal deletion. This is an example of genomic imprinting. Now, what is the importance of genomic imprinting? Still, a lot of research is being carried out. People are not sure what is its role in development, but research has so far shown that it takes parents of two opposite sex, of course, to produce a healthy embryo. And this embryo, when it develops, the male genome, the 23 chromosomes that are coming from the dad, actually are controlling the placenta development, while the female genome, those 23 chromosomes, are contributing more to the embryonic development. So placenta development more controlled, greater control by the male, the paternal chromosomes, while embryonic development more tightly controlled by the maternal genome. See if we missed something. No, I think we're done. Yes, we are done with this lecture. Let me know if you have any questions. No questions? How was this lecture? Did you guys understand it? Okay, I see 3 thumbs up. So we are going to end this meeting today. As usual, I'll post the recording. Please do not miss your deadlines. I can see that there's a quiz coming up. There's also the homework 1 deadline tomorrow. So turn in your assignments on time so that you don't miss easy points. So I'll see you tomorrow with a new chapter. Oh, I see a question. What is the quiz on? I think there's a study guide for the quiz, Jenny, and it would usually involve in the discussion section. You would have discussed this with you on quiz 1, and that's why I want you to go over the study guides. It is all the topics that we've covered so far, mainly conceptual questions. Clear? Okay, thank you.